The cofactor F420 plays an important role in the primary and secondary metabolism of many prokaryotes. Measuring this molecule in environmental samples enables a deeper understanding of its prevalence and function.
This technique enables the analysis of the cofactor in difficult sample materials such as sludge and soil. Thereby, the lysis of the material and pre-purification before analysis are the central steps.
The protocol includes different steps during the sample preparation. These steps must be planned and prepared very well before starting with the sample processing. For example, the preparation of fresh buffers is important.
To begin with sample lysis, place 5 grams of the sample in a 50 ml conical tube. Then add 5 ml of 2 times lysis buffer to the samples and bring the volume to 10 ml with distilled water to reach a final concentration of 0.5 grams per milliliter. Vortex the diluted samples for 20 seconds, followed by autoclaving for 30 minutes at 121 degrees Celsius. For dry samples like forest soil, bring to a final volume of 20 ml with distilled water after autoclaving and vortex the diluted sample again for 20 seconds as demonstrated. Once the samples cool down, centrifuge the sample lysate for 5 minutes at 11,000 times G and collect the supernatant. Prepare a 5 ml solid phase extraction or SPE column filled with 100 mg of weak anion mixed mode polymeric sorbent by conditioning the anion exchanger with 3 ml of methanol. Then, equilibrate the anion exchanger with 3 ml of distilled water. Load up to 9 ml of the supernatant from the centrifuged lysate onto